Hello everyone, welcome to PD's Research Bites, and in this video I am going to talk about COVID vaccines. The purpose of this video is to provide an overview of types of coronavirus vaccines. But first let us look at the headlines. These are some of the names of the vaccines we see commonly in the news. Pfizer-BioNTech COVID-19 vaccine, Covaxin Covishield, Moderna, Oxford AstraZeneca vaccine, Sinovac. I have selected the most popular ones. There are so many other vaccines also available. So vaccine contain the same virus or bacteria that cause disease. So coronavirus vaccines could be either killed, otherwise it is called inactivated, or weakened. The other name is live attenuated, or part of the virus could be used in vaccine production. Protein is used, subunits could be used in vaccine production, or it could be from the genetic material of the virus. So let us see how the vaccine works. All vaccines are designed to teach our immune system to fight a disease, to recognize and block the virus or the pathogen by producing antibodies that protect us from disease. It is safer for our immune system to learn through vaccination than by catching the disease. The specialized memory cells prime the immune system to produce cells and antibodies to target the virus. So this is the coronavirus, and the brown part is the genetic material, the outer covering. There is spikes, you can see the spike protein, the red color projected spike protein. So using our ACE2 receptor, the spike protein enters our cell. So in the cell, it releases the genetic material and viral proteins are formed, the virus is released. So our immune system responds to the viral protein and the virus. So there are a lot of cells get activated, and there are several mechanisms involved in the immune response, like T cells, they are the killer cells. So helper and the killer T cells can destroy the virus. There are macrophages, B cells which produce the antibodies. So our immune system responds to the virus. So there are different types of vaccines in development, and the goal of most of the vaccine is to get the target antigen. The antigen here is the spike protein of the coronavirus, so that our body creates immune response and produce antibodies. So let us see the types of vaccines one by one. So the first type I am going to discuss here is the inactivated whole virus vaccines. So from the name itself, it is clear that inactivated whole virus is used, or otherwise it's the killed coronavirus is used in this type of vaccines. So these type of vaccines use inactivated whole virus, so it doesn't cause disease but still generate an immune response. The examples are Covaxin by Bharat Biotech India, Sinovac and Sinopharm from Chinese pharmaceuticals. So inactivated virus, this is the killed coronavirus, and when it is injected to our body, it is recognized by our immune system and our immune cells get activated, antibodies are released, and then we get protection by triggering the immune response of the body. So the advantages and disadvantages of inactivated whole virus, they are relatively simple to manufacture, relatively stable, well-established technology. We do have other existing inactivated vaccines, including the whooping cough, rabies, hepatitis A vaccines and influenza vaccines using the same technology. It is suitable for people with immunocompromised systems, no live components or no risk of the vaccines triggering the disease. Booster shots may be required. So let us see the next type of vaccines. These are protein-based, protein subunit vaccines. These vaccines contain proteins, either whole protein or protein fragments from the coronavirus which are recognized by the immune system to trigger a response. Example is Novavax. It is still under research, not yet released. So protein vaccines using the spike proteins. The vaccines are developed using the spike proteins, and when it is injected to our body, our immune system recognizes these proteins and then create an immune response by triggering the immune cells and producing the antibodies, and then several mechanisms are involved for the protection. So the advantage and disadvantages of protein-based vaccines. Again, well-established technology is used here. Hepatitis B vaccine is a protein-based vaccine which is used since so many years. Suitable for people with compromised immune system, no live components or no risk of the vaccine triggering the disease. It is relatively stable, but it is complex to manufacture. Determining the best antigen combination takes time. Adjuvants and booster shots may be required. Adjuvants in the production of vaccines and booster shots may be required for these type of vaccines. So moving to the third type, viral vector vaccines. So from the name itself, it is evident that we need a vector for this type of vaccines. So in this, a harmless virus, example adenovirus from chimpanzee, is altered by introducing a part of coronavirus. The adenovirus is like a vehicle to carry the coronavirus genetic code. Here the code for COVID-19 spike protein. So adenovirus is the vector here. That triggers an immune response in our body. Example, Oxford AstraZeneca is a viral vector vaccine, and Covishield is also viral vector which is produced in collaboration with this vaccine, and Sputnik vaccine from Russia is a viral vector vaccine. So this is coronavirus. The genetic material is separated from the virus and then it is introduced first to the vector. This is the adenovirus vector we are talking about, and then it is injected to our cell, and then the spike protein of coronavirus is produced. As soon as the spike protein is produced, our body will react by producing antibodies, by activating all our immune cells, and mechanisms will get activated. So that's about the viral vector vaccines, and let us see the advantages and disadvantages. It creates strong immune response which involves B cells and T cells, well-established technology. We have Ebola vaccines using the same technology, and previous exposure to the vector could reduce effectiveness, not the virus, exposure to the vector, relatively complex to manufacture. Let us move on to the genetic vaccines or the nucleic acid vaccines. RNA, DNA vaccines it's called. These vaccines use genetically engineered RNA or DNA to generate a protein that itself safely prompts an immune response. COVID-19 vaccine uses a synthetic version of the virus messenger RNA. That is why it's called mRNA vaccines. So when these vaccines is injected, our cell starts building the relevant viral protein. For example, the protein here is COVID-19 spike protein. Then our immune system get activated. The examples of this type of vaccines are Pfizer-BioNTech vaccine and Moderna vaccine. So coronavirus here, and the genetic material is separated and then injected to our cell by using gene gun, or a carrier molecule is used here, and then the spike proteins are generated which will create an immune response by activating the immune cells and the antibodies. So the advantages and disadvantages: no live components or no risk of the vaccine triggering disease. Immune response involves B cells and T cells, relatively easy to manufacture, but some RNA vaccines require ultra cold storage. Never been licensed in humans. Booster shots may be required. So this type of vaccines, it is a newer type of vaccines. There are no other existing vaccines which is used by using this method. But ongoing research in infectious disease such as HIV, Zika virus is going on, and it is also, this technology is also used in cancer and allergy, but it is all under research. So two other type of vaccines which is under production and not yet released and not that popular, but they are live attenuated virus. It is using a weakened type of coronavirus, and also another category of vaccines which is using virus-like particles. So that's all in today's video. So these are some of the reference. And if you like the video, please share and subscribe. And hopefully I will come up with another video on vaccine efficacy. It's a very difficult topic. We have to look at the research around the vaccine efficacy and then the trials and the results and things like that. So hopefully I'll meet you soon. Goodbye. Thank you.